Hello everyone, thank you for watching this video. Today we are going to talk about statistic power and resolution. You perhaps have learned much more about engine sequencing knowledge in recent days. You might be working hard to make use of those variants found in your sequencing projects. One of them is doing association analysis. I will introduce some information which are related to assess results from your association analysis. Hope you can get some ideas to improve your experiment design or optimize your analysis. Any question is welcomed. Email or Slack both okay. Let's start from statistic power. Statistic power was a possibility for us to reject null hypothesis which states no effects for marker on the trait. Peter in 2017 introduced a method that could be used for power calculation. Resolution, that is how small the regions are, but we don't have a criterion to say how small is good or how big is not good. It depends on different projects. Many factors have effects on statistic power and resolution, including accuracy in phenotyping and genotyping. You already have many ways from last courses to estimate the genotyping quality. But the following major issues are commonly discussed. They are effect size, allele frequency, sample size, and population structure. Effect size was defined as phenotypic variance within the population, explained by the SNP you are looking at. From the figure, we can find different effect size have different power, and this is a big difference. In short, higher effect size increases power a lot given the sample size. If the potential causal variants have less effect size, how should we do to detect signal? We can assume that one SNP only have an effect of 5%. Statistic power was calculated at a different sample size from 200 to 400 and to 800 samples. We can find a bigger sample size increased power. It looks good, but one thing that this will also increase the cost in your project, especially when phenotyping or genotyping are expensive. From this slide, we can find the statistic power was correlated with allele frequency positively, which also means that it's more difficult to detect a significant signal for SNPs with mean allele frequency lower 1%. That is because very few people will carry these alleles and cannot result in genome-wide significance. This figure only showed SNPs with a frequency from 0 to 0.05, so the power displayed here are not very high. If frequency increases continuously, the power will also increase. In practical GWAS projects, alleles with a frequency lower 1% will be removed. Now let's talk about the confounders. Confounders are also very important factors which you always detect more or less in your project. Confounders include the population structure and the environments. These two factors are very important issues that could lead to severe false positives. They have to be corrected if detected. We will focus more on population structure. The source for the population structure is the difference in the genetic background. This figure shows a theory for population structure. In both case control groups, they have several populations, but these subpopulations have very different allele frequency and loci, which is not causal one. But their allele frequency difference fit the group well. GWAS based on this dataset will result in many false positive signals, although true positive signals could be also included. We have an example to explain population structure more. Look at the years in peaks. We could assume the ear shape as a binary disease in Yorkshire population. Ears are eruptive and small. In landrace, they are bigger and floppy. However, by setting one of them as control and another as case, the GWAS will detect much more loci which are not really associated with ear shape. That is because the two populations have very different genetic background. One is from England and selected for good growth performance. Another was bred in Denmark, selected also for growth performance but more famous for higher reproductive performance. They also differ in meat quality, feed efficiency, and body conformation. If we did GWAS on this dataset, it's very likely your results will look like this messing. Many markers go up above the threshold. You don't know which are true or false. A better way to map this gene is to build a resource population where the two populations were used as parents to produce F2 segregating offspring. Here, the F2 samples share more in genetic background. Linkage analysis or GWAS in F2 will become more powerful. From the last example, it is easy to know there is a population structure according to breeding records. But sometimes we have no idea about the population structure until we get the results. This is very common as current GWAS always involves thousands of individuals, which inevitably include the population structure. How could we detect this and how could we correct them to remove those false positives? We will calculate inflation factor, also called lambda. Lambda was used to represent the extent how population structure is distorting your results. The logic behind this method is that apart from a small number of SNPs that show a true association with the trait or disease, the test statistics for other SNPs should follow the distribution under the null hypothesis of no association between a SNP and the trait. However, artificial difference in allele frequencies due to population structure or genotyping errors will affect all SNPs, so the test statistics will be inflated across the whole genome. Inflation factor fluctuates between 0.95 and 1.05. We will trust this association analysis, but for some meta-GWAS studies, which include thousands or millions of individuals, we also accept a much bigger lambda, for example 1.4 or 1.5. We also draw a QQ plot to check the results. In QQ plot, SNPs without effects on trait will drop on the line. Associated SNPs will deviate from the line. In left figure, it is good, because many SNPs that shows no effects on the trait, they drop on the line. Only a very small part of SNPs which are true causal SNPs, they are deviating from the line. In the right figure, it is not right, because almost all points deviate from the line, and this is not true. I have to say the population structure is not that easy to remove. In this example, 5 causal loci or QTL were stimulated. In figure A, population structure was not corrected. Markers on every chromosome were found to be significantly related to the trait. But we know this is not true because we only have 5 causal SNPs. In figure B, after correcting the structure, most false positive signals were suppressed. However, one false positive was still there and one new false negative signal showed up. This means after correction, we lose one true causal SNPs, but keep one false positive. QQ plot will show many false positives that are deviating from the null hypothesis if there is no correction, but we are not but this figure not shown here. So, do we have other factors still affect our results? As mentioned in first slide, accuracy of phenotyping and genotyping also affecting our results. Try all your best to get precise phenotypes and genotypes, especially the phenotypes. Researchers are still developing other statistic models to improve power and remove confounders. So, what should we do to increase the power? For effect size, it is biologically determined. Not much to do. For low frequency, there are already some studies which were using specific mating system to increase the allele frequency. Once this allele frequency increased, it becomes the common variant. More samples would increase power, but cost should be also considered. Population structure is the main issue in most GWAS projects. It has to be tested and corrected. In linkage analysis, select families showing big phenotypic variation. Now let's see the resolution in association studies. People always expect to do a fine mapping and locate one or two genes as candidates to be explored in further studies. However, the mapped regions are always either big or containing many genes. Therefore, we have to do extra jobs to narrow down mapped regions. Marker density and recombinations are two important factors that should be considered to improve your resolution. In GWAS, linear mixed models always assume markers that are independent from each other. The association was done in variant traits model. From the figure, we can find that more markers give us more opportunities to approximate the causal genes. The red flag is the causal gene, the green point are SNPs, color blocks are genes, intergenic regions are not shown. In linkage analysis, this is a little bit different. Linkage analysis always use sparse markers to do the first round mapping with poor resolution. But it doesn't matter because the fine mapping by screening more variants in mapped regions will be carried out. From the figure, we could find that if markers in a region are linked with each other, genotyping one of them is enough to detect this region, which contains the causal gene. But still, increased marker density will be helpful. For linkage analysis, there is another efficient way to improve the resolution. That is to accumulate more recombination events in your population. In this figure, P0 is the parents. They have divergent haplotypes, the blue one and the red one. And many recombination events were detected in F2. If we stop here, we are able to do fine mapping by linkage analysis, although these long haplotypes will result in poor resolution. But if we continue maintaining the populations to get more generations, say go further to F7, much more recombination events would be detected and they will split those long haplotypes into shorter ones. Linkage analysis will pick up those shorter haplotypes regions and regions out, and this time it is a better fine mapping than that in F2. This is also why we say the biological basis for linkage analysis is recombination. Okay, that is all for this morning. We can go to Zoom meeting room for questions. Thank you.